Hello, in this video we will discuss about the influenza virus life cycle as well as the pathophysiology. So there we will discuss about their mode of action here. So first of all we should need to understand here is the influenza virus. So this contain of bilayer of phospholipids, the plasma membrane like structure, and contains spike protein as well as this spike protein is also known as the we will discuss here. It is also known as the hemagglutinin, HARH. And here you can see this is the phospholipid bilayer, and the black color is showing that is the neuramidase enzyme. So here is the this is the trimeric hematoglutanin. This is trimeric basically. It is contain three subunits. So three subunit. So here is you can see this is the RNA polymerase which contain RNA. So it is the RNA virus contain RNA. So there are many type of RNA for synthesis of protein by the using of machinery of host. So here is the influenza common life threatening a virus in the United States. Each year death rate 20,000 persons in each year. So that is the basically is the life threatening, but mostly it can be recovered. So here is the it infect the upper respiratory tract because the upper respiratory tract contain the receptor for the endocytosis of this virus. So the virus are present in the air, and during the inhalation time the air contain a droplet of water which contain a virus, and it can infect the person to person, and this person to person infection due to the trimeric hemagglutinin hema hemagglutinin. So here is the viral cell entry through a hemagglutinin we will discuss in this video here. So let's begin to understand first of all it's a symptom. So this here is a droplet with virus. So the symptom can be coughing, sneezing, fever, and nausea. Even the high fever can be occur due to the immune response and increase the temperature for killing this virus, because the normal function will not available in the 37 degrees Celsius, so the temperature will be rise for killing it. So here is the upper respiratory tract contain this receptor is known as the sialate receptor. This sialic acid. So the sialic acid is the receptor for it. So the sialate receptor are present in the virus. So it is receive the sialic acid. So here is the fusion peptide. This is the fusion peptide, one of three subunits. So it is three subunits, and here I am drawing just one for understanding. So this is the hydrophobic and buried within the core of the hemagglutinin trimeric complex. So in this way, the trimeric complex hemagglutinin, which that's contain this uh, hemagglutinin. So here you can see the black color is showing which that is attached, and the fusion peptide will be occur. So here is the fusion peptide. In this way, here is the inside the glycoprotein polysaccharide containing sialic acid in the terminal of the polysaccharide. And in this way, the here we will discuss about the sialic acid first for understanding. With attachment with the galactose is the basically is the polysaccharide which that is attached. So this is known as the glycoprotein. And after the glycoprotein with binding with the glucose galactose formation. So the galactose will attach with the carbon number 2 to the carbon number carbon number 3 to carbon number 3 to carbon number 2 of the sialic acid. So here is the sialic acid, while the galactose is the terminal region for the attachment of the sialic acid. So the sialic acid will bind with the alpha or 2 to 3 in the avian or birds, while the alpha 2 to 6 linkage in human upper respiratory tract just only. So that is why the infection site is the upper respiratory tract will be damaged and infected. So in this way the alpha 2 to 6 glycosidic bond are present. So let's begin to understand their mode of action and their entry. So after the binding with the sialic acid with the sialated receptor, so the cleavage will be occur from the hemagglutinin. So the cleavage formation will be fusion peptide will be released after the cleavage. So after the cleavage, the fusion peptide, three fusion peptide subunit will be cleaved. So here I am trying first one for understanding. So let's here is the endocytosis will be occur after the binding, and after the binding the cutting of the hemagglutinin, which that is after cutting to endocytosed, and this is known as the endosome, and it is the endosome formation will be occur into the cytoplasm of the upper respiratory cell. So the after cleavage hematoglutanin is here looking like this. While here is the blue color is showing that is lysosome. This lysosome endocytosed into the endosome, and after this it contain a hydrogen ion, and it will increase the pH. After this increasing of this pH to create a different mechanism. Here you can see the uh, number one is the trimeric subunit of the hematoglutanin will be released and move down with the attachment with the plasma membrane of the endosome, and this receptor binding region back, and in this way it attached with the endosome with a virus, endosome plasma membrane with a virus, and after the detachment of the hematoglutanin, his hair is looking like this, and in this way the hematoglutanin will relax and release from the receptor sialic acid, while the attachment through a fusion peptide. So the fusion peptide will be attached with the phospholipid bilayer. After this attachment, here is you can see this entry of the proton is create this reaction due to the lower pH, due to the increase of the proton into the endosome. After the increasing of the proton will decrease the pH. After the decreasing of the pH, this all three mechanisms will be occurring this manner. So the peptide fusion peptide here fusion peptide is the red color is showing in the figure, which that is basically uh, uh, here, and will bind with the phospholipid bilayer of the endosome and the receptor binding region back. In this way the receptor binding region will be back of the hemagglutinin. So in this manner, the increase of the pH will attachment of the hematoglutanin with the phospholipid bilayer. So important thing is that. So let's discuss about here is you can see in this diagram to understand that, and in this way the hematoglutanin molecule will be basically the receptor binding region back, it is made up of hematoglutanin. So the hematoglutanin molecule will be separate from the receptor from the sialic acid. So in this way, a number of trimeric hematoglutanin molecule in the same region of the membrane mediate fusion between the viral and host membrane. After the fusion with the hematoglutanin, so this is the hematoglutanin molecule, the receptor binding region will be relaxed. So after this binding to open this outflux, uh, uh, basically exocytosed of the RNA. So this exocytose of the RNA is the major target for the releasing of the RNA of the virus into the cytoplasm is very important, and this importance is due to the it will go this RNA polymerase RNA polymerase containing strands, which that is the basically 8 strands containing RNA polymerase go into the nucleus and bind with the our DNA, host DNA. After the binding of the host DNA, basically the transcription will be occur to produce messenger RNA many more. After this, many more messenger RNA will be produced, which that go into the rough endoplasmic reticulum where the ribosomes are present. This ribosome is the machinery of the synthesis of the protein, is known as the translation from the messenger RNA. So from the messenger RNA produce the protein. This protein is very important to produce the spike protein, which that is basically the matrix protein and trimeric hematoglutinin, as well as the neuroamidase enzyme. So this the phospholipid bilayer is also produced. So the phospholipid bilayer is basically is not a part of the translation, but it is come from the cytoplasm of the host cell. So the RNA plus polymerase will be produced. This is the basically is the after the transcription and translation to produce this enzyme, and this on the other hand the protein will produce the receptor, this hematoglutamine receptor, and also the fusion peptide will be formed, three fusion peptide. So here I am drawing just for understanding, but this diagram is not quite too much creative, but you can understand as well. So the plasma membrane of the upper respiratory tract is here, which that is damaged due to the exocytosis of the of the virus by a packaging. So the packing to budding and budding to the exocytosis. After the exocytosis, the membrane of the upper respiratory tract cell will be again bind, but in this way the much more using of the phospholipids bilayer due to the synthesis of the more virus will damage the upper respiratory tract and to increase the immune response. So let's begin to understand the immune response, but first of all we should need to understand the type of RNA. So after the release is here, so after the releasing, this virus will be released and infect again to another cells, but here is the PB2, PB1, PA and HA and NP, NA, M and NS. This is the type of RNA. So this is the viral RNA. 8 viral RNA is showing in the figure. So let's begin to understand important thing is that the immune response and how it's possible. So here is the upper respiratory tract contain MHC class 2 molecule, which that is show as an antigen representing cell to represent the antigen, which that is a piece of the virus will donate to the with the binding with the CD4 T lymphocyte is the helper cell after the binding recognition to produce the chemokines and cytokines. These cytokines basically is the inflammatory mediators to create the inflammation as well as recruitment of the B cell for the production of antibody and plasma cell for the production of antibody and B memory cell as a seasonal B memory cell, and again infection create again seasonal. So here is another thing is the cytotoxic T cell which kill the upper respiratory tract cell, and the macrophage recruitment and innate and adaptive immune response will be occur. So in this way, that is the influenza virus mode of action and the symptom of the disease, and this can be